Um, I'm glad you were able to join today. We're really excited to have a guest presenter, Dr. Carol Boss, and I'll introduce her to you. She's a cancer research advocate, a health educator, a medical writer, and a biomedical engineer. She has a PhD in biomedical engineering from Texas A&M. She became an advocate following her diagnosis with breast cancer in 2004, and she's volunteered for way more groups and organizations and programs that I could mention here, but to highlight it is she is an ECOG Akron advocate, so she knows what the NTCG advocates are about. She's also a member of the IRB at Baylor and serves on the External Advisory Board for the Simmons Comprehensive Cancer Center. She's also one of the four founding editors of a new scientific journal called Convergent Science Physical Oncology. And it focuses on how experimental and theoretical science contributes to a better understanding of cancer complexity as it evolves in the patient, and in the development of more effective diagnostic and treatment strategies. So a wonderful new journal that is available. Previously, Carol served for 5 years as the National Advocate for the Physical Sciences Oncology Centers at the NCI, and she has worked with cross-disciplinary teams at 12 different cancer centers across the U.S. to develop comprehensive cancer advocacy programs. So she's had a lot of involvement in many different areas, a strong background. She's a friend and a consultant for CISN. So thank you so much, Carol. We're really pleased to have you today as our presenter. CISN's mission is, again, to build bridges, and that's why we're doing this webinar today. And the developers, the web content actually was developed by myself, Diana Chingos, Sandy Feinstone, and Kimberly Jackson-Warren. And we did this several years ago. The PDF that I sent out was the original one. Carol has made a lot of really wonderful improvements to it. So the talk you hear today will be different than the one I sent out. And if you would like the newest version, I would be happy to resend that out for you because it is much improved. We had a copywriter who went through the website, and then the PowerPoint was put together by myself and Carol. So Carol, why don't you begin? Thank you.
Sure, and thank you so much for the opportunity to be able to present. So our topic today is understanding precision medicine. So I want to welcome you to this webinar and thank you for taking the time to learn more about this topic. So, next slide, please. And just to make sure that we're all looking at the same slide, since I'm not looking at the screen, you should be looking at learning objectives. Is that correct?
Yes.
Okay, then we're synced. All right, so we all know how difficult it is to receive a cancer diagnosis and to make treatment choices, including those for targeted therapy and for tissue donation request. So we believe that the more people understand the issues that surround precision medicine, the more comfortable they'll be with discussing this information with their medical team, and the better able they will be to make decisions about their own health care. Next slide. So under training overview, these 4 topics will give you a good understanding of different aspects of precision medicine. Please remember that this is a complicated and it's a rapidly evolving field, and although the outlook appears promising, it's important to understand the challenges that are involved. Next slide. Precision medicine and you. In this section, we will discuss what precision medicine is and how it relates to you. We'll also define a few background terms to help you better understand the topic. We now recognize that cancers are fundamentally diseases of the genome, and that understanding cancer begins by identifying the abnormal genes and proteins that confer the risk of developing cancer. Identifying and analyzing these abnormalities will define how we diagnose cancer, how we determine or determine how we develop and use targeted therapies to treat cancer and to shape the strategies we use to prevent cancer. So after decades of research, we're now poised to enter a new era of medical practice where detailed genetic and other molecular information about a patient's cancer is routinely used to deploy effective, patient-specific remedies to treat it. We're now about to enter the era of precision medicine. Next slide. So, what is precision medicine? The National Institutes of Health defines precision medicine as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. Next slide. Precision medicine is a medical model in which treatment decisions are tailored to the individual patient. The promise of precision medicine is that cancer treatment for each person will be as individualized as the cancer itself. It provides accurate predictions about a person's susceptibility of developing a disease, the probable course of that disease, and the patient's response to treatment. With precision medicine, we will have the ability to identify an individual's possible side effects to a drug. For example, does this patient have an increased risk of developing specific adverse effects with this drug treatment? Also, the ability to predict an individual's response to a drug. Will this drug be effective for this patient? And finally, the ability to use a biomarker that can diagnose, treat, and monitor this individual's cancer. So what is precision medicine? We are all zebras. When you hear hoofbeats, think of horses, not zebras. I think we've all heard that expression. That was said originally by Dr. Theodore Woodward, who was a professor at the University of Maryland School of Medicine. And he told his medical interns that phrase in the late 1940s to teach them the art of diagnosis. Now with precision medicine, we see the zebra in all of us. We focus not on what makes us part of the herd, but what makes us unique. Precision medicine uses each person's unique combination of genetic, clinical, and environmental information. Every person is different in multiple ways, including what they eat, the types of stress they experience in everyday life, their exposure to environmental factors, and their genes. Many of these play a role in health and disease. Next slide. So what exactly is the difference between personalized medicine and precision medicine? And forgive me if during this talk if I switch up the two, because I think a lot of us do. There's a lot of overlap between the two terms. According to the National Research Council, which is the working arm of the U.S. National Academies, which includes the Institute of Medicine, personalized medicine is an older term, but some people, including me, still use the two terms interchangeably. There was a concern that the word personalized could be misinterpreted to imply that treatments and preventions are being developed uniquely for each individual. But as we've discussed previously, in precision medicine, the focus is on identifying which approaches will be effective for which patients based on their genetic, environmental, and lifestyle factors. So the National Research Council therefore preferred the term precision medicine to personalized medicine. So that's what we'll be using. Next slide. So you've probably all seen a lot about precision medicine in the news. Headlines trumpet the promising advances in the field as well as the failures that reflect the evolving nature of the field. The top quote was from just a few days ago. Precision medicine is often more complicated than a lot of people say. The second headline, which was from earlier in the year, was a happier one. The hope is that every disease has molecular underpinnings that once understood will lead to new treatments or even cures. Next slide. Making it personal. So as we know, every person is different in many ways, including their DNA. Personalized medicine is predictive. It uses state-of-the-art molecular and diagnostic tools to precisely predict an individual's health risk and outcomes and involves the patient in those discussions. It's preventive. It places an emphasis on wellness and prevention to prevent or mitigate disease, taking into consideration how much a patient is willing or able to do. It's preemptive. It can include an action plan for patients, a prescription for ongoing good health. It's personalized. The plan must take into consideration every aspect of a person's life, from what foods they like to whom they live with and who they are responsible for. And finally, it's participatory. It educates individuals about how much they have control over. It empowers individuals to determine how much they're willing and able to do. Next slide. To understand the field of precision medicine, we must first know some terminology. What is a biomarker? The NCI defines a biomarker as a biological molecule that's found in blood, other body fluids, or tissues that is a sign of normal or abnormal process or of a condition or disease. It may answer questions that deal with prognosis. Am I likely to develop this cancer? Diagnostics. What type of cancer is it? Predictive. Is this the best treatment for my cancer? Pharmacodynamics. Is this the optimal dose for me? Reoccurrence. Will the cancer return? Biomarkers are also called molecular markers or signature molecules. Next slide. Background information. We aren't there yet, but the ideal tumor marker would be a blood test in which a positive result would only occur if cancer is present. It would be wonderful if the test also indicated the stage of the cancer and its response to treatment. An added bonus would be if it could also identify the optimal treatment for that patient. But tumor markers are not always reliable for the following reasons. Normal cells as well as cancer cells can make most tumor markers. Tumor markers may also be associated with non-cancerous conditions. Tumor markers are not always present in early stage disease. And even when tumor marker levels are high, cancer may not be present. Remember that tumor markers that may be helpful in monitoring some cancer patients during and after treatment most often are not reliable in detecting cancer. Next slide. Background information. Here are some more definitions that you'll need to know. Genetics is the study of heredity, or how the characteristics of living things are transmitted from one generation to the next. Genomics is the study of genes and their function. Genomics examines the molecular mechanisms and the interplay of genetic and environmental factors in disease. The word epigenetic literally means in addition to changes in genetic sequence. It is used to refer to any process that alters gene activity without changing the DNA sequence. For decades, scientists assumed cancer was caused by an irreversible damage to a critical stretch of DNA. But there is emerging evidence that some cancers are caused by epigenetic changes, tiny chemical tags that accumulate over time and can turn genes off. Experiments show that epigenetic changes, unlike mutations, can be reversed. Next slide. Another term that you'll hear frequently is pharmacogenomics. Pharmacogenomics is the study of how genes affect a person's response to drugs. This relatively new field combines pharmacology, which is the science of drugs, and genomics, the study of genes and their functions. to develop effective, safe medications and doses that will be tailored to a person's genetic makeup. For certain drugs, pharmacogenetics may help to determine whether patients are rapid or slow metabolizers, which refers to how long the drug stays in your body, as well as whether they are responders or non-responders, which means whether the drug works for you. Having the ability to predict a person's individual response to a drug, both in terms of therapy benefit and the likelihood for adverse events, will play a crucial role in preventing the use of a drug that is likely to cause serious effects for a patient without benefit, and guiding the selection of optimal therapies that are most likely to benefit the patient. Next slide. So, as you can tell from the visual, genes are the recipe of the cell. They contain all of the instructions for assembly. Proteins are the product of these recipes. Although protein biomarkers hold great promise, there are significant challenges to consider. The vast number of proteins that exist in the body can make it difficult to identify and describe them. Proteins are continually moving and undergoing changes, and proteins exist in a wide range of concentrations in the body. Next slide. Metabolomics is the study of the metabolic responses to drugs, environmental changes, and disease. It's an extension of genomics, which is the study of genes, and proteomics, which is the study of proteins. As you can see from the image, metabolites provide a closer marker to how cells are functioning than genes or proteins, and they have the potential to impact clinical practice. Metabolomics is sometimes used to understand how inherited genetic differences among people lead to differences in how they metabolize drugs. Proteomics and metabolomics are still in their early stages of study and are not yet used routinely in the clinic, but they do possess great potential. Next slide. Precision medicine today. As shown in the diagram above, early detection testing includes risk assessment, prevention, and targeted monitoring. Although early detection testing will continue to be based on large population risk, the hope is that new tools will enable screening tests such as mammograms and colonoscopies to be more accurate and to find disease earlier when it's easier to treat. Diagnosis. Fulfilling the promise of personalized medicine requires that these findings be translated into precise diagnostic tests and targeted therapies. Therapies. New molecular tools may also provide doctors with information on which drugs their patients may or may not respond to, and/or the likelihood of their developing adverse effects associated with certain agents. Finally, response monitoring. To truly wipe out cancer cells within the body, it's not enough to have effective drugs that target some of the cancer growth pathways. It is important to have a way of monitoring the cancer itself so that drug therapy can be adjusted should the tumor change or evolve. Next slide. The overarching promise of precision medicine is to optimize the medical care or outcomes for each individual. It recognizes that the best prevention strategies, the best treatments, medications, and dosages may differ from person to person, resulting in customized patient care. Precision medicine takes into consideration that all human diseases have both molecular and environmental components. As the uniqueness of individual tumors become clear, it's now understood that even the right combination of drugs will fall short as resistance develops and responses are not durable. Science must now be able to distinguish mutations that are drivers of the disease and therefore targetable from those that are merely present. Personalized medicine is working on this now. Next slide. In his State of the Union address this year, President Obama announced that he's launching the Precision Medicine Initiative, which is a new research effort that will impact how we improve health and treat disease. The President's budget for fiscal year 2016 included $216 million in funding for the initiative for the NIH, the NCI, and the FDA. The budget includes $70 million in increased funding for the NCI to advance the field of precision oncology. Next slide. Here are some specifics on the plan. The Precision Medicine Initiative has both short-term and long-term goals. The short-term goals involve expanding precision medicine in the area of genetic research. Researchers at the NCI hope to use this approach to find new, more effective treatments for various kinds of cancer based on increased knowledge of the genetics and the biology of the disease. The long-term goals of the Precision Medicine Initiative focus on bringing precision medicine to all areas of health and healthcare on a large scale. The NIH plans to launch a study involving a cohort of at least 1 million volunteers from around the U.S. who will provide genetic data, biological samples, and other information about their health. These data will be used by researchers to study a large range of diseases, with the goals of better predicting disease risk, understanding how diseases occur, and finding improved diagnosis and treatment strategies. The plan was shaped by a 2011 report from the National Academy of Sciences Expert Committee that urged the federal government to pour money into a new taxonomy of diseases that would define them by their molecular and environmental causes rather than their physical signs and symptoms. Next slide. With additional resources in its fiscal year 2016 budget, the NCI will increase funding to support clinical trials that offer two distinct approaches to advancing precision medicine. The first approach recruits patients with all types of cancer and then selects a targeted drug based on the specific genetic abnormalities of the patient's tumor. The NCI is using this approach in its Molecular Analysis for Therapy Choice, which is known as NCI MATCH program, which enrolls pediatric and adult patients with tumors that no longer respond to standard therapy. The NCI plans to scale up efforts to identify genomic drivers in cancer and to apply that knowledge in the development of more effective approaches to cancer treatment. NCI MATCH supports the overall initiative because it's an example of how the NCI will accelerate the design and testing of tailored treatments for cancer by expanding genetically based clinical cancer trials, exploring fundamental aspects of cancer biology, and establishing a national cancer knowledge network that will generate and share new knowledge to fuel scientific discovery and to guide treatment decisions. Next slide. The second approach, which is demonstrated in the NCI Lung MAP study, recruits patients with one type of cancer and then subdivides patients into genomically defined subsets, treating each subset with a different targeted drug. So LungMap, which is SWOG S1400, is a multi-drug, multi-sub-study, biomarker-driven squamous cell lung cancer clinical trial that uses state-of-the-art genomic profiling to match patients to sub-study testing, investigational treatments that may target the genomic alterations or mutations that are found to be driving the growth of their cancer. Both types of trials have important and achievable goals: making the molecular characterization of cancers the clinical standard for accurate diagnosis and treatment, identifying or developing an array of treatments that can be matched to the molecular features of a tumor to successfully control the disease. Another feature of these trials is the opportunity to link their genomic findings to clinical data, lifestyle risks such as tobacco use and obesity, environmental mutagens, and viral infections. This additional feature will become a resource to support further discovery and to identify new strategies for cancer prevention, early diagnosis, and early intervention. Next slide. There are many challenges to implementing precision medicine, and some of them are listed on this slide. For example, direct-to-consumer genetic tests are not designed to provide a clear-cut answer as if one is pregnant or not pregnant. The information received from genetic tests can be extremely complex and is best understood with the help of both doctors and genetic counselors who know you, your medical history, your family history, and other crucial details. Stakeholders in the implementation process include diagnostic and pharmaceutical companies, governmental and regulatory bodies, payers, physicians, and patient groups. Next slide. Tissue donation provides the very foundation of precision medicine, and patients play a key role in donating tissue to research. There is no other way to achieve precision medicine without research on patient-donated tissue. Next slide. Tissue, or biospecimens, is material from your tumor, hair, or body fluids. The tissue can be normal or cancer tissue. Tissue can be used for testing for individual diagnosis if a patient has a genetic signature known to benefit from a drug. It is also used for research to learn about genetic variations that characterize a risk factor, response to treatment as a responder or a non-responder to a particular drug, or the risk of side effects. Next slide. Here are all of the categories of research for which tissue acquisition is essential, as well as some examples of some successes. Next slide. Tumor tissue is routinely biopsied and analyzed to confirm or rule out a cancer diagnosis. A pathologist performs such analysis on fresh tissue, frozen tissue, or tissue that has been fixed in formalin, which is a preservative. The pathologist then performs the histological examination. Leftover tissue can be donated for research. Next slide. Opportunities to donate tissue. In most cases, tissue remains after diagnosis and can be donated. Clinical trial donation. Patients enrolled in clinical trials are increasingly receiving requests for tissue in the informed consents for their studies or as a separate consent addendum. These studies may require tissue samples to help identify the genetic or protein targets of the investigational drug, or to determine the characteristics of individuals who respond best or who do not respond to the research agent. Such studies are referred to as correlative studies. For obvious reasons, many patients find it easier to donate blood, urine, saliva, and hair for research. However, the need is greatest for solid tumor tissue, particularly for rare cancers or rare subtypes of common cancers. This can be obtained by biopsy. Next slide. So case study is the Komen Tissue Bank. An example of a tissue bank is the Komen Tissue Bank at the IU Simon Cancer Center. It was established in 2007 with the support of the advocacy community, which has been instrumental in procuring tissue donations. It's the only repository in the world for normal breast tissue, which is crucial to our understanding of breast cancer. The Komen Tissue Bank collects fresh frozen tissue, formalin-fixed paraffin-embedded tissue, and blood products, including whole blood, plasma, serum, and DNA from lymphocytes. Next slide. Confidentiality is a big issue. Donated tissue can be linked to a patient's data. Tumor tissue can be identified, meaning that the hospital or research center will be able to link the information derived from the material to a specific patient. In such cases, researchers can access information about the patient's diagnosis, treatments, and outcome. Donated tissue that is stripped of any identifying data is called de-identified. It is useful, although no outcome data is attached to it. Some research takes place without patients' knowledge. With a pathologist analysis of a tissue specimen in the cancer diagnosis, the anatomic pathology laboratory information system transmits the diagnosis, which is a reportable event, to the hospital cancer registry and the state cancer registry. The state cancer registry creates a record of the patient's cancer in the cancer registry record. Next slide. Factors affecting tissue quality. The life cycle of the tissue can be divided into 4 stages: collection, processing, storage, and analysis. Collection refers to how the tissue is obtained from an individual. At this stage, there are multiple factors that can affect the biomarkers that are present in your tissue. Called pre-analytics, these factors can have an effect before the tissue is processed, stored, or analyzed. Second stage is processing. Once the tissue is collected from the body, the next step is called processing. This refers to the handling of the tissue to prepare it for testing or storage. The third stage is storage. Tissue is often stored in large facilities called biobanks that may be located within hospitals, research centers, or private facilities. The fourth stage is analysis. Tissue testing or analysis also needs to be standardized to enable accurate comparison of results. Next slide. The phrase informed consent refers to the process of communication between a patient and a physician to elicit the patient's understanding of a proposed medical intervention and that results in the patient's authorization or agreement to undergo a specific medical intervention. That's a definition from the American Medical Association. An informed consent document should include the following items: what types of data will be collected and how the data will be used and stored, the patient's willingness to be contacted about the use of his or her biospecimens and/or data in future research studies, any benefit or potential risk to the patient's family, and his or her community, such as whether there's a risk of stigmatization or discrimination that are based on the research results, whether individual or aggregate research results will be released to the patient, the family, or the provider, and if they are, the mechanism for communicating these results, the type of research that will be conducted, such as the specific cancer type, or looking at all possible disease conditions. The informed consent should also include the possible benefit to the investigator or the institution and information concerning potential conflicts of interest. And finally, how the patient can discontinue participation and withdraw consent. Next slide. During this section, we will discuss ethical issues. Next slide. It is important for newly diagnosed cancer patients and their family members to understand the ethical implications of personalized medicine. Although many patients may currently benefit from research that has already identified targeted therapies for their specific cancer types, this field is relatively new, and much work is still in progress. Therefore, patients may need to consider the following questions. If available, Should they have a genetically based test to determine the possibility of benefiting from a specific targeted therapy? If a patient is diagnosed with a cancer associated with known inherited genetic mutations, such as BRCA1 or 2 gene in breast cancer, ovarian cancer, and other cancer types, should family members receive genetic testing to help determine their risk? More questions include, should patients agree to donate their tissue? It's important for cancer patients to understand the implications that such decisions may hold for them and for their family members. Next slide. So although precision medicine holds great promise, several public policy and ethical challenges exist that must be addressed before it can fulfill patient needs safely and effectively. All stakeholders must be actively engaged in overcoming these challenges. They must work together to carefully study the multiple ethical, legal, and social issues raised by this research to help fulfill the promise of precision medicine while ensuring patient protection and preventing any misuse of new genetic technologies and information. Next slide. The promise of precision medicine. The overarching promise of precision medicine is to optimize medical care and outcomes for each individual. Precision medicine takes into consideration that all human diseases have both molecular and environmental components. It recognizes that the best treatments, medications, dosages, and preventive strategies may differ from person to person, resulting in customized patient care. Next slide. The reality for patients. Genetic information is biologically linked to individual identity, making personalized medicine an intimate and a serious process. Although the promise of improved outcomes is real, there's much more information about each person's genomic makeup in their medical record. What patients need to understand. If patients consent to donating their tissue, they should have a full understanding of how their genetic biospecimens and data will be used, including what research will be conducted now and in the future, what benefits may result from the research, how the information will be used and what information will be revealed about participants who donated their tissue for this research, and which entity owns the donated tissue. It's crucial for patients to ensure that they receive a clear, thorough explanation during the consent process and that they ask questions. Next slide. Ethical challenges. History has shown that genetic information can be used for good and for bad. There are a range of ethical concerns relating to precision medicine, including privacy concerns and discrimination. Next slide. There are other important ethical issues. Biobanks are used to process and store collections of human biological tissue specimens and related health data. These biospecimens are important in increasing our understanding of the many aspects of disease. As noted previously, there's numerous ethical, social, and legal considerations that must be taken into account concerning biobanks that include informed consent, confidentiality, secondary use of sample data over time, return of results, and data sharing. For direct-to-consumer testing, as we discussed previously, traditionally, genetic tests have been available only through healthcare providers, such as physicians, nurse practitioners, and genetic counselors. Healthcare providers ordered the appropriate test from a laboratory, collected and sent the samples, and interpreted the test results so that patients would receive the support they needed when their results were delivered. This is changing. Maybe we can talk more about this in the discussion. Next slide. So, ethical issues summary. As an evolving medical model, precision medicine promises to offer safer and more effective cancer treatments by matching drugs to a person's individual information at the molecular level, the individual's molecular fingerprint. So though the promise of personalized medicine is exciting, there's several associated ethical considerations that must be resolved. Next slide. Molecular diagnostics. In this section, we will discuss the role of molecular diagnostics in precision medicine. Next slide. Background. So the human body is made up of trillions of cells that talk to one another to keep you healthy. Normal cell growth requires a communications network that functions properly. This network is an extremely complex system of protein pathways. The genetic changes that are associated with cancer result in changes in proteins that disrupt the cell's communication network. The challenge once cancer is diagnosed is to locate these rebel genes and proteins that have hijacked normal cellular communication and correct the problem before it becomes life-threatening. This requires analyzing the molecules inside the cell using molecular diagnostics. Here you'll see the definition of genomics and proteomics, which we discussed earlier in this talk. Next slide. So simply put, molecular diagnostics includes all tests and methods used to identify a disease or the likelihood of developing a specific disease by analyzing biomarkers, such as DNA, RNA, or proteins. Standalone tests do not accompany a drug, but they look instead for specific gene mutations. Biomarker tests look for specific protein markers that may indicate the presence of cancer. And companion diagnostic tests are developed along with a drug to find which group of patients will benefit from that specific drug. Next slide. So here are a few examples for the categories that we discussed on the previous slide. In addition to well-established biomarkers, such as EGFR in colorectal cancer and lung cancer, and HER2/neu and ERPR in breast cancer, many new biomarkers are being investigated in multiple tumor types. As for companion diagnostics, pharmaceutical companies are now increasingly focused on developing new agents that can target only cancer cells and providing companion diagnostics to identify those patients who may benefit most, those who are unlikely to benefit, and/or those who are at most risk for adverse effects. Next slide. Importance. The image above shows the many areas in which molecular diagnostics may be used. This spans the entire scope of cancer care, beginning with risk assessment and moving through to surveillance after diagnosis and treatment. Although the traditional pathological examination of cancer remains an essential clinical tool, newer technologies such as microarrays, mass spectrometric proteomic analyses, and protein chips are being used more often, although not yet routinely used everywhere or for all cancer types. Next slide. Potential advantages. In the future, your doctors may be able to predict your response to chemotherapy or radiation based on your molecular signature or expression signature. This could eliminate much of the clinical guesswork when deciding among multiple therapies that have been approved for a specific cancer type. Next slide. Potential challenges. Challenges include higher cost for some of the targeted treatments, added cost to the diagnostic test, lack of availability across the country, the need for standardization of tissue and the validation of the test. We need electronic health record systems, and we need genetic counselors. As an evolving medical model, precision medicine promises to offer safer and more effective cancer treatments by matching drugs to a person's individual information at the molecular level, that individual's molecular fingerprint. Though the promise of personalized medicine is exciting, there's several associated considerations that must be resolved, which we just discussed. Next slide. In the future, at least two current trends will make personalized health care even more important than it is today. The first is, we now know that on a molecular level of every cancer is different, not only between different tumors, but even between different areas within a single tumor. This means that we need to match the right drug to the patient who we know will respond best to the drug at the right time during the course of treatment. Second, as we develop more and more effective lines of therapy for patients, we will use personalized healthcare even further to decide on when to switch therapy. Patients may undergo routine biopsy during their course of treatment, even if non-invasive methods like blood-based diagnostic tests become reliable and feasible. So in the future, patients will have their tumors profiled not only for genetic drivers, but also for predictive immunotherapy markers at different time points in their course of treatment. Next slide. Remember, the goal of molecular diagnostics is to provide the right drug for the right patient at the right time. Medical personnel need training on how to discuss precision medicine as well as how to provide decision aids to assist patients to handle all of this new information. Next slide. Summary of precision medicine. Remember that this field is changing rapidly, and precision medicine and molecular diagnostics may revolutionize how we diagnose, treat, and follow cancer. Next slide. This slide illustrates the research process, bench to bedside through clinical trial participation. New treatments, including targeted therapy, must be tested in clinical trials, and patients are needed to volunteer to be in a trial. Patients are a critical piece in the research process, and patient tissue donation is critical at all stages of the work. Next slide. So in summary, precision medicine, including the molecular diagnostic tests that provide information to your healthcare providers, may ultimately revolutionize cancer prevention, diagnosis, treatment, and follow-up. Although there are many pathways involved in most tumors, making targeted therapy more complex than originally thought, this emerging approach continues to offer great promise, such as in the following: the ability to make more fully informed medical decisions, a higher probability of desired outcomes, reduced probability of negative side effects, increased focus on prevention and prediction of disease, earlier disease intervention than previously possible. But it is important that all stakeholders' needs are met. Healthcare providers desire easy-to-access, easy-to-use molecular testing services.
So Carol, are you finished? So Mark, it looks like. Hello, I think somehow we lost Carol. Patty, are you still there?
Yes, I'm still here. I was
Oh,
muted.
okay. Well, luckily we are at the end of the talk.
Yeah.
Okay,
So
I'm here. Can you hear me now?
yes, now we
Yeah.
hear you.
Okay.
And I just moved to the disclaimer slide. So why don't you finish up anything you'd like to say?
Oh no, I'm done. So I was finished. So
Oh, okay, very, very good. Well, thank you so much. Thanks to Carol, who gave that amazing presentation, to Patty, who helps us all the way through, to Jane and Sandy for helping us pull together the web content, and for our funders. So I'm going to unmute people, and if you have a question, just feel free, give me a moment here to unmute. I'll let you know when I'm at it. Takes me a little moment because I have to hand do it. Okay, everybody is unmuted, so if anybody has a question for Carol, please go ahead.